Hello, BNG319 class. Welcome to the recorded lecture for this Tuesday, the 7th, where we're going to talk about metabolism. This will serve as sort of an introduction to all things metabolism, and there's a lot to cover, so we'll get going. But first, one of you had a muddiest point from last Thursday regarding Neuraminidase and Tamiflu, and you remember me talking about Tamiflu as an inhibitor of the Neuraminidase enzyme, Neuraminidase being key in the release of viruses like the flu virus, as well as some other viruses. So what happens is the virus will replicate itself inside our cells, and then it will bud and come to the surface. And as it comes to the surface, it attaches to a receptor, and that receptor contains a sugar-type molecule called sialic acid, and the neuraminidase will cleave off that sialic acid, and the sialic acid itself is attached to another protein called hemagglutinin, which all it does is bind to that sialic acid. And the neuraminidase will cleave off that sialic acid so the virus particle can be released and go on to infect new cells. If we inhibit the neuraminidase with Tamiflu or some other kind of anti-flu drug that is molecularly similar to sialic acid, the neuraminidase active site will be blocked, and thus it will not act on the cell surface sialic acids that are present on these receptors, and the virus will not be able to be released. It will just hang out on the cell surface until macrophages come and eat it up. And if there's no virion release or no viral particle release, you get much less viral replication, and then hopefully that will alleviate your flu. Okay, so first of all, why? What's going on here? So the virus attaches to cell surface sialic acid, and the surface of our cells are decorated with all sorts of oligosaccharides that have different structures, and these are some stylized structures here, some different glycans that can either be attached to lipids in the form of glycolipids, or they can attach to proteins, membrane proteins, that essentially are either attached at the amino end of the side chain of asparagine, or an oxygen of a serine or threonine amino acid. And they stick out into the extracellular milieu. And you can see they often have repeating units of different sugars: galactose, mannose, N-acetylglucosamine, N-acetylgalactosamine, etc. Very often their ends are capped with sialic acid, which looks like this. It's also called N-acetylneuraminic acid, hence neuraminidase. Sialic acid, it's all the same thing. Alright, so I mentioned that drugs like Tamiflu will inhibit the flu neuraminidase competitively. So they'll bind at the active site. And you can see that Tamiflu and other ones look a lot like the structure of sialic acid, with some key differences here. You know, it's a lot more hydrophobic on the end here for Tamiflu. You've got a phosphate group down here attached, and an aminophosphate group, to be clear, as well as this double bond right here instead of an oxygen here. So there are a variety of differences between sialic acid and Tamiflu for sure, but they are structurally similar enough that certainly this will bind to the active site of the neuraminidase. And like I said, sialic acid is found on the surfaces of cells, either in glycolipids or in cell surface oligosaccharides that are attached to membrane proteins. Tamiflu will bind, it will inhibit the neuraminidase enzyme, this viral enzyme, preventing virus from replicating. And so how does that happen? Well, you can imagine that people have mapped, groups have mapped the interactions between viral neuraminidase and a sialic acid, which you can see here to the left, and they've also mapped binding of the neuraminidase to the Tamiflu, which you can see here to the right. So if we look at what's going on here, we have basically this is the neuraminidase active site here. We are complexed by a bunch of different arginines, which are positively charged amino acid. You can see they are interacting with a carboxylic acid group, so this is negatively charged. And so this isn't super surprising. You've got positively charged amino acid side chains in the active site interacting with a negatively charged carboxylic acid right there. So that is holding that part of the molecule in place. You've got glutamate and aspartate right here, acidic amino acids. One of them is creating hydrogen bonds with this OH. And again, we have arginine here that's performing some kind of interaction with this carbonyl oxygen here, as well as we've got some tryptophan isoleucine here that are just kind of providing the shape for the binding site so that sialic acid can bind. But then you've got things like glutamate that are making some sort of interaction, usually hydrogen bonds here with this terminal OH group on the sialic acid here. And you can see some key differences in Tamiflu. You can see that we're not having any binding over in this area where we have an arginine 224 isoleucine. They are binding, probably more likely interacting with this alanine 246 and isoleucine 222. We still have this arginine 371 that can make a interaction. This arginine 292 could also possibly make an interaction. These three arginines could make an interaction. You can see they're making an interaction with the regular sialic acid. They can also do that potentially with Tamiflu here, as it's shown. And here we have an aspartate, which might possibly be making an interaction here with this amine. And we have arginine making, this is arginine 152, and you can see this is making some sort of interaction with sialic acid. It could potentially make the same kind of interaction with Tamiflu. So there are differences here. There are differences in the way this thing could interact, but in general, a lot of the same amino acids are interacting with Tamiflu in the binding site of neuraminidase that would interact if this was sialic acid. But it is different enough that it's going to bind Tamiflu to the exclusion of sialic acid. And then we bind all those up, and the viral neuraminidases can no longer cleave off the sialic acid on the cell surface, allowing viral particles to be released. And so this is the key thing that is happening with drugs like Tamiflu, that are these homologs, or these structural homologs anyway, of different cell surface sugars. They are basically being fed these fake substitutes here in order to keep the actual activity of infecting cells down to a minimum. Okay, so before we get into metabolism, I'd like to make a couple of announcements. We'll have a quiz on Thursday when I get back. That quiz, I've decided, is only going to cover what we talked about last week with enzyme kinetics and enzyme inhibition. We won't talk about metabolism. We won't deal in metabolism on this quiz. I'll save the metabolism for next week's quiz, which will be November the 14th. If you have muddy points regarding the quiz on the 9th, please send me an email. Unfortunately, I don't think I am going to be able to do a voluntary recitation this coming up week or this week. We will do a voluntary recitation for the metabolism quiz, hopefully on Monday the 13th. Okay, so we talked a little bit. We began to talk about, when we talked about kinetics, when we talked about reactions and enzymes and their inhibitions, we talked about pathways. And the hallmark of a metabolic pathway is that the product of one reaction becomes the substrate of the next reaction. So you have a bunch of reactions in series. You start out with a substrate. The first enzyme converts it to its product. That product becomes the substrate of the next enzyme, which converts it to its product. That product becomes the substrate of the next enzyme, and then you're basically down the line, you end up with your end product. So the product of one reaction becomes the substrate of another. And what this does is it produces our key products and intermediates in living cells like our own, and bacterial cells, yeast cells, whatever, you name it, any living cell. So for example, here we have a linear pathway with which we can make the amino acid serine from this 3-phosphoglycerate, which is a molecule that is found in glycolysis, which we will see a little bit later. Some pathways are differently shaped. They're not linear reactions. They actually occur in a circular pattern, where the product of one reaction becomes the substrate of the next, so forth and so forth and so forth, until we're basically back at the beginning, and then we start all over again. And that's what the TCA cycle is all about. And we'll talk more about the TCA cycle in a little bit. So these pathways can take a variety of shapes depending on what's going on. Okay, so here we can see the beginnings of the breakdown of glucose. And glucose, for a lot of us, either whether we're humans, animals, even plants to some degree, even bacteria and yeast, will break down glucose as part of glycolysis. And so we phosphorylate glucose, we reduce it, or we oxidize it rather, we reduce NADP, and then we linearize it, and then we go from there through glycolysis. And so that essentially starts a lot of processes, the breakdown of glucose. So the sum of all reactions that can take place in a living thing is called metabolism. A lot of it, especially if you're eating glucose, glucose breakdown, glycolysis, becomes sort of the focal point to which all of these other things happen, like syntheses. So we synthesize proteins, we synthesize DNA, we synthesize our cell membranes. If you're a bacterium and you're eating glucose, you use some of that carbon to synthesize your cell wall, etc., etc. And there's a lot more things that get synthesized, like cofactors, or rather coenzymes and things like that, that get made. And what do we do to synthesize these things? Well, we break down food, and that can take the form of sugars like glucose, proteins, etc., 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 lipids, you name it. And these are all food for specific organisms. Okay, so metabolic pathways, again, they all look unique and different depending on what's there. But they're just a bunch of letters, arrows, numbers, whatever, right? You've got a linear pathway that goes from A to E. Here's a spiral pathway. It's got a bunch of A's. And here's a circular pathway that goes A to E as well. It is true, there's a bunch of letters, numbers, and there's always arrows showing the directionality of the pathway. but they all have a purpose. Everything has a purpose to denote how molecules are broken down and or synthesized by organisms, whether those organisms be us, or those organisms be E. coli, or Saccharomyces cerevisiae, or mice, or crabs, you name it. So these all have a purpose to tell us where are the nutrients going, where are the elements going, like carbon, nitrogen, oxygen, where are things going from food to building blocks and macromolecular structures. And so on the whole, metabolism is pretty complex, and here is sort of a schematic of general metabolism based on a lot of metabolic pathways that have been studied in a variety of different organisms, and they're color-coded to denote, like this green here is lipid metabolism. You've got carbohydrate metabolism in the blue. TCA cycle you can clearly see here. Things that are called secondary metabolites over here. And so there's a variety of things. And these pathways have been studied in one or more organisms. And they kind of make up the whole of metabolism. Some organisms have a lot of these things. Some don't have certain things. It all depends on on what the organism is and what it does, where it lives, what it eats, that sort of thing. There's a lot of things that are involved in determining metabolism. But it can encompass a lot of things, and that makes it useful in a way, especially if we want to alter metabolism for any reason. We'll talk about this on Thursday, but if we want to alter metabolism to make products that we can do something with, that we can use for alternative syntheses, that we can package and sell as drugs or other useful compounds, we can take advantage of cellular metabolism a lot and maybe add pathways, alter pathways, and things like that. We can sort of put our own stamp on this general metabolic map of an organism and make it do some things based on what has been learned over the course of the last 5 or so decades regarding metabolism. Okay, so let's zero in. Let's put a little more of a fine point on it to talk about reactions and how metabolism is essentially set up, with a bunch of reactions. And they're associated, these reactions, as you may know, with free energy change. And so you have spontaneous and non-spontaneous reactions. And if you remember from general chemistry, and if you remember even from stuff I believe I've talked about early on in class, you have spontaneous reactions where the delta G, or the Gibbs free energy, is less than 0, and the non-spontaneous reactions, which the delta G is greater than 0. And so, of course, by the name, the spontaneous reactions will happen. They'll be spontaneous. And the non-spontaneous reactions won't happen unless they're coupled to something that's spontaneous. And the coupled reactions, the spontaneous reaction is providing the energy that's needed for the non-spontaneous reaction. And this is true in metabolism. You have a spontaneous reaction, and one of the key ones is the hydrolysis of ATP to ADP in phosphate. That releases 7.3 kilocalories per mole of energy. Energy that can be used to run non-spontaneous reactions, like this amination of glutamate to make glutamine. This is in general a non-spontaneous reaction, but if we couple it to ATP hydrolysis, we make an overall spontaneous reaction, which allows us to make our glutamine, and we have a little energy that gets left over, most likely wasted. But regardless, we've accomplished what we want to accomplish in making glutamine by this pathway here. So like I said, ATP tends to be the great coupler. There are other hydrolyses that will be coupled to non-spontaneous reactions to make a spontaneous reaction. But ATP is the critical one, and it's the one that's used most often. And it's hydrolyzed in many pathways. And you look at a metabolic pathway, you can often find some energy-consuming steps where ATP is hydrolyzed. And then in some pathways, ATP can be produced because we have to recycle that ATP in order to keep the cell moving, to keep those non-spontaneous reactions going by coupling them to available ATP. So we can produce ATP. This is a reaction 1,3-bisphosphoglycerate, which is hydrolyzed to give us 3-phosphoglycerate plus phosphate. And that has a delta G, a highly negative delta G in terms of metabolism, of negative 11.8 kilocalories. That's enough to be able to recharge ADP and phosphate to make ATP. And so we can see this is plus 7.3 kilocalories. So easily we can couple these two reactions to make our ATP again. Okay, so when we do this coupling, the hydrolysis of bisphosphoglycerate with ATP synthesis, the net reaction and the net free energy, we have negative 11.8 kilocalories plus 7.3 kilocalories. We get this water on both sides, phosphates on both sides. Here's our net reaction, which is 1,3-bisphosphoglycerate plus ADP yields 3-phosphoglycerate plus ATP with our delta G of negative 4.5 kilocalories total. Okay, so here we ask the question. Here we have a step that's early in glycolysis. It is a couple reaction right now involving the transformation of fructose 6-phosphate to fructose 1,6-bisphosphate. And here it is on the top here. The first reaction, not the fifth reaction, the first reaction is spontaneous. True or false? Well, we look at the delta G here, and we know right away what the answer to that is. We look at delta G and we say false. The first reaction has a positive delta G, positive 3.9 kilocalories. So the first reaction is not spontaneous. In fact, that's why it is coupled to ATP hydrolysis, which is very spontaneous, with this negative 7.3 kilocalories of delta G. So what is the net free energy change of this coupled reaction? We have the makings of fructose 1,6-bisphosphate, and we have the hydrolysis of ATP. One is 3.9 kilocalories, the other is negative 7.3. We do some excruciatingly simple math to get a delta G of the coupled reaction of negative 3.4 kilocalories. Our coupled reaction is indeed spontaneous with its negative delta G. Okay, and so here is an overarching look of some of the goals of metabolism here. We have different metabolites which serve as building blocks, like amino acids, which serve as building blocks for proteins. Fatty acids serve as building blocks for phospholipids. Carbohydrates, monosaccharides serve as building blocks for phospholipids, for polysaccharides, and even DNA and RNA. And nucleotide nitrogen bases serve as building blocks for DNA and RNA. So everything is connected, and in a lot of cases, everything is interconnected. So when we think about nucleoside triphosphates, those NTPs, we know why they're important. You've got ATP. ATP hydrolysis is basically the energy that runs a lot of cellular reactions, or certainly a lot of cellular pathways. And we have ATP, GTP, UTP present, and CTP present in RNA, as well as DNTPs that are present in DNA. And so they're important for a variety of reasons. So these, needless to say, are very important biomolecules. They also are, or can be, part of the structure of some of our oxidation reduction coenzymes. Namely, we talked about NAD, NADH. We talked a little bit about NADP, NADPH. And of course, there is also FAD and FADH2. All of these can act as a receptor for electrons and a proton or two to either oxidize or reduce some other molecule. So NAD will be converted to NADH if we're oxidizing a molecule. NADH will be converted to NAD if we are reducing a biomolecule. And these are all done enzymatically. And so just a reminder of what we've talked about last week, we have our coenzymes NAD, NADH. Here is what they look like, and you can see you have an ATP basically attached to this whole thing, but the business end is right here, and the reason it plays a role in the oxidation and reduction of substrates is because you have this oxidized form which can pick up electrons plus hydrogens and it'll become a reduced form, and it will act as a reducing agent for whatever molecule it has picked up the hydrogens and electrons from. Okay, and you can see an example of this, a good example. There's several examples, honestly, in the pathway, the TCA cycle, or the citric acid cycle. And you can see as we go around the pathway here, there are at least 3 incidences where NAD is required to act as an oxidizing agent. Here we're going from isocitrate, which has this structure, to alpha-ketoglutarate, which has this structure. And you can tell, if you look here, you can tell that we have, well, first of all, we've gotten rid of a carbon dioxide, but we have also oxidized the molecule. We're taking this OH and turning it into a carbonyl. And to do that, we've used NAD, and it has become NADH. The NAD has been reduced. We can take alpha-ketoglutarate and turn it into succinyl-CoA by first adding a CoA as well as taking NAD and oxidizing it again. We have an oxidation here that gives us another carbonyl. Over here, it's maybe a little more clear to see. We've got 2 C4 compounds. We're not coupling anything with the loss of a carbon dioxide. We have 2 C4 compounds. You can easily see what NAD is doing here. NAD is oxidizing malate to oxaloacetate and becoming NADH. And you can see we have a hydroxyl here is becoming a carbonyl right here. So this is where we've oxidized the molecule. And this has been done enzymatically, of course, with NAD as the coenzyme. FAD is also getting involved as a oxidation reduction coenzyme, and you can see that here it's being done in a different way. We're taking a carbon-carbon single bond and creating a carbon-carbon double bond from succinate to fumarate, and FAD has done the job on that one, and it's been reduced to FADH2. So this is oxidation and reduction done in metabolic pathway, and most metabolic pathways may involve some kind of oxidation reduction in order to get from reactants to products here. Certainly TCA cycle is probably the best known of those things. And let's not forget we have group transfer agents like acetyl-CoA, and again we see the ADP nucleotide is part of this molecule. Here is the coenzyme A with the thiol here. That will soon become, if we react out the hydrogen here for an acetyl group, we have acetyl-CoA, and this acetyl group can be transferred from one molecule to another depending on what the cell is trying to do, and the whole CoA molecule will then go back to being coenzyme A once the acetyl group has been transferred to, say, a fatty acid or something like that. So overall, we look at the general scheme of metabolism. We have 2 kinds of things. We have catabolism, we have anabolism. What's going on here? We have large molecules and small molecules. Sometimes we want to break down large molecules. Sometimes we want to make large molecules. Obviously, things like DNA, RNA, protein, those are large molecules that we want to make. Large molecules like starch and other foods, proteins that are foods, we want to take them and make them small and use them to make energy as well as building blocks. So converting large molecules to small molecules is called catabolism, and that will usually allow us to recharge our ATP. So converting ADP plus phosphate to ATP. Making things, anabolism, will take small molecules to make large ones. It usually requires the hydrolysis of ATP. Where do we get all that ATP? Where does it come from? Well, if in our cells, it comes from the mitochondria. In bacterial cells, it comes from the the cell cytoplasm and the cell membrane. So for our example, we have the mitochondrial matrix, where we have this setup here, where we have an electron transport chain, and we're pumping protons or hydrogen ions. We're pumping them up a gradient, and we're creating this gradient where protons will go down the gradient, and that will provide energy to recharge ADP plus phosphate to make ATP again, using this complex, this protein complex called ATP synthase. So usually what happens, we get 2 free hydrogen ions, 2 electrons, half of an O2 is also going to give us H2O. So that is a byproduct of this whole thing as well. So where does it get made in eukaryotes? So it's mostly the mitochondria. And you can see this mitochondrion has a funny shape. It's got this outer membrane, kind of looks like a bacterium in a way. Then it's got this matrix here, which has a lot of space in it. It's got these 2 membranes. It's got these very weird looking inner membrane that has all these folds and things like that, and a lot of space, and who knows why that's even there. So it's the powerhouse of the eukaryotic cell. Why does it have such an complex interior shape with all these folds? The answer to that can be found in the placement of the electron transport chain in the ATP synthase. Essentially, these folds provide more surface area. More surface area gives us more space to put more of these complexes, the electron transport chain and the ATP synthase complex. So the more folds the mitochondrion has, the better able it is to make a lot of ATP when needed. So I said metabolism is divided into 2 parts. One is catabolism, where compounds, big compounds, are broken down into smaller parts. And usually with that, energy is released. And then there's anabolism, where larger compounds are biosynthesized from smaller ones, or what we call building blocks. And usually an input of energy is required. Let's first touch on catabolism. And catabolism starts with food, whether it's our food or whether it's bacteria food. Bacteria food could be just glucose. For us, our food are things like hamburgers, steaks, broccoli, that sort of thing. And our food is more complex, obviously, than bacteria food, in that, it contains proteins, carbohydrates, fats, triglycerides, things like that. And all of these things can be broken down to their components, like amino acids from proteins, monosaccharides from carbohydrates. Triglycerides can be broken down into fatty acids and glycerol. And then these things can go into further catabolism, where they get converted to pyruvate, to acetyl-CoA, and then they will enter the citric acid cycle. The reduced coenzymes in the electron transport chain and oxidative phosphorylation will result in the production of ATP from the food we eat. And so what does this look like? So we have things like starch. There are enzymes in our mouth, in our saliva, that will break down starch into oligosaccharides, glucose, and then there's undigested polysaccharides that won't be broken down in our mouths. They'll go through the stomach, more or less unchanged. They hit the small intestine, and they get broken down again further, where some of these oligosaccharides and undigested polysaccharides become monosaccharides by the action of amylase and other enzymes that break down poly and oligosaccharides. Triglycerides don't really get broken down in the mouth. They get broken down in the stomach by lipases that can tolerate acidic pHs. They get broken down into diacylglycerides, fatty acids. Undigested triglycerides can then go to the small intestine, where again lipases would work to make more fatty acids, glycerol, etc. Proteins don't really get broken down again in the mouth. They get broken down in the stomach, where things like pepsin, if you remember from pepsin when we talked about its enzyme activity and its conditions that make it optimal for its enzyme activity, it has an acidic pH optimum, and so that's because it's from the stomach, and it'll convert proteins into oligopeptides, amino acids. Some undigested polypeptides get through, and then they get further broken down in the small intestine to more of their requisite amino acids. And so the question, what do you suppose about the enzymes that perform these catabolic processes? Well, any like amylase that comes from the mouth, from saliva, is probably going to perform best at neutral pH. Stomach enzymes like the stomach lipase and pepsin is going to operate best in acidic conditions with acidic pHs. The enzymes like amylase, lipase, trypsin, chymotrypsin that are in the small intestine would operate best when there's a basic pH. Okay, so here's starch hydrolysis. Amylose is in starch. It is a polysaccharide that's in a lot of the starch we eat. It gets hydrolyzed to make glucose, many glucoses. Do we remember what type of glycosidic linkage it is here that holds amylose, the sugars and amylose together? It is an alpha 1,4 linkage, so that as carbon 1 is attached to carbon 4, and alpha because it is pointing down, right? So what would an enzyme called? And we've already talked about such an enzyme. What would an enzyme be called that breaks down amylose? Well, that's a simple one. It's amylase. Again, like these enzymes are named basically by what they do. So an amylase breaks down amylose. And my guess is because it breaks down, it's called amylase and it breaks down amylose, that it has a very a rigid specificity for amylose. Lipid hydrolysis gets performed by lipases, where fatty acids are removed, and you're left with basically glycerol, maybe some monoacylglycerides, but they take triglycerides and fats, and they break them down into fatty acids, glycerol, etc. Protein hydrolysis, again, the peptide or the amide bonds in protein get broken. You end up with individual amino acids, and all over in your stomach and your intestines, you have proteases, peptidases that will hydrolyze proteins at certain amino acids to give you individual amino acids that can then be either further broken down or they can be incorporated into your proteins. And as you'll see later, we need to do a lot of that because there are certain amino acids we do not make that we need to get from our food. Alright, so everything essentially centers on glycolysis. It is an important metabolic pathway. Basically, it's taking glucose and turning it into what we call central metabolites, so things like acetyl-CoA, pyruvate, that sort of thing. And what this does is it produces a net of 2 ATP molecules for glucose. And this is just from glucose to acetyl-CoA. And in our cells, it's connected with the mitochondria in that acetyl-CoA goes over the mitochondrial membrane, and the TCA cycle happens in the mitochondria here. And so basically one glucose is converted into 2 pyruvate. We get 2 ATP molecules as well. And so that is just the beginning of, basically it's just the beginning of energy conversion here, but it is just the beginning of metabolism, because we have now just entered what we call central metabolism. And so here's the net reaction. 1 glucose yields 2 pyruvates, or 2 pyruvic acids, because in the cell, what form is it going to take? Is it going to be pyruvic acid, or is it going to be pyruvate? Probably, if you remember from fatty acids and remember from your amino acids, that this likely has the pKa that says it's going to be deprotonated under physiological conditions, which will be pH of around 7. So we'll see it as pyruvate, not as pyruvic acid. So at physiological pH, pyruvic acid exists in its conjugate base form, or its deprotonated form, i.e. pyruvate. Okay, so here is glycolysis, all stepped out right here. We have about 10 steps, and they can be broken down further. Steps 1 through 5, where we have glucose, and we make glyceraldehyde 3-phosphate. They're often known as the preparatory phase. Steps 6 through 10, where we take glyceraldehyde 3-phosphate and turn it into pyruvate, are often known as what we call the payoff phase. Why do you suppose that is? Well, I'll tell you. It is essentially because the preparatory phase, we are using energy to convert and prepare the carbon substrates for further catabolism, to eventually give us and to pay us back the energy we used here and more. So we're preparing the substrates to come to the second half of glycolysis, where, as you can see, we are making ATP again here at step 7, and also here at step 10. And you say to yourself, we look at step 1, we look at step 3, we look at step 7, we look at step 10. You say, wait a minute, Dr. Brigham, didn't you say, I know you said this, didn't you say that you get a net of 2 ATP in glycolysis, turning glucose into pyruvate? I swear I heard that. I'm going to sue. And then I will say to you, hold on a second. You have glucose, and you have C6 compounds, because glucose has 6 carbons. Eventually here, when we get to this aldolase step, we convert a C6 compound, this fructose 1,6 bisphosphate, into 2 C3 compounds, dihydroxyacetone phosphate, or glyceraldehyde 3-phosphate. And they can interconvert. So essentially we've gone from 1 mole of glucose here and 1 mole of fructose 1,6 bisphosphate here to 2 moles when we're at step 6 here of glyceraldehyde 3-phosphate. So everything, the NAD, ATP, all of this stuff, you must multiply by 2. So essentially we're losing 2 ATP here in the preparatory phase, and we are gaining back 2 ATP here. We are gaining back 2 ATP here in 1 molecule of glucose. So that's where the net 2 ATP comes from. What happens once you've made pyruvate? Well, there's a variety of things. It depends on what you are, and it depends on what you're doing. So in yeast, pyruvate, it can be converted into ethanol. And you can see it gets converted into acetaldehyde, which then gets reduced to make ethanol. In an anaerobic environment, and this happens when you're exercising, this happens in your muscles, pyruvate is reduced to lactate in your muscles anaerobically when you do a lot of exercise. And that's why your muscles cramp up and feel achy at the end of exercise sometimes. You've got a lot of lactate in there. In the mitochondria, Pyruvate is converted to acetyl-CoA by decarboxylation and attachment of coenzyme A plus an oxidation to give us acetyl-CoA. And then acetyl-CoA goes on to bigger and better things. And so, if you remember the big map from of all the metabolism that's known in the world, there were a lot of pathways that intersected with each other. So much like highways intersect and have junctions and things like that, metabolic pathways can share intermediates and have junctions at certain intersections that in metabolism speak are called nodes. And many pathways will share intermediates with glycolysis, which is important to know. And that is often why we call glycolysis and the TCA cycle central metabolism, because that's basically the center of activity. That's where everything kind of springs off of here. So let's look at that. So a lot of things can feed into glycolysis. Glycolysis is essentially glucose being metabolized, but we can also feed into from galactose being metabolized, mannose and fructose being metabolized. And then of course there's the pentose phosphate pathway, which we will see on the next slide, will also feed into intermediates of glycolysis. And so here's the pentose phosphate pathway, which again, like glycolysis, can be broken into two parts, where we have the oxidative reactions that aren't reversible, looking at glucose 6-phosphate making 6-phosphogluconate, making ribulose 5-phosphate, and then into the non-oxidative reactions that are reversible. And this is where we can start to look at some of our 5-carbon compounds like ribose and xylulose, and those are important, of course, for making nucleotides. You can see, here is one of the intermediates, ribose 5-phosphate. That can then go on to nucleic acid biosynthesis. So that can go on to make ATP or ADP. It can also go on to make either DNA or RNA nucleotides. So when we talk about directionality, and I've talked about basically reversible versus irreversible, we look at the direction of the arrow in a metabolic reaction equation. I hesitate to call them equations because we shouldn't really be setting things equivalent to each other. This just indicates a direction. So in this reaction, what does the arrow represent? This is taking glucose 6-phosphate and making glucose and phosphate. It's hydrolyzing that phosphate group. So it's the direction. This is the direction, kind of like I just said. The catalyst, the enzyme, is taking glucose 6-phosphate as our substrate. This indicates this is substrates to products. And this goes in the direction of glucose 6-phosphate to glucose and plus phosphate. And so here, we need to recharge ATP. We can do that in the mitochondria using that ATPase that I showed you that's in the mitochondrial membrane. And we can do that by coupling it to another reaction. So here's a reaction for recharging ATP. And this one is positive 7.3 kilocalories, whereas ATP hydrolysis is negative 7.3 kilocalories. This is, to recharge it, is positive 7.3 kilocalories. Is there a reaction that when coupled to the below reaction, this reaction, would result in an overall spontaneous reaction? The answer obviously is, of course, there is a reaction that will do this. And here it is. Here's 1,3-bisphosphoglycerate plus H2O. Basically, it's the hydrolysis of 1,3-bisphosphoglycerate to make 3-phosphoglycerate plus phosphate. The delta G there is negative 11.8 kilocalories. And you can see easily that if we coupled these two reactions, we end up with a spontaneous reaction overall. A net spontaneous reaction. And so there is, obviously there's a lot of times when we are going to eat glucose and break it down, glycolysis, in other words, there are times when, maybe not so much when but when organisms want to make glucose. And it could be that you're eating a non-carbohydrate carbon source, like lactate, glycerol, amino acids, whatever. And you still need to get into the realm of the sugars. And so when you're making glucose, is it essentially, do we just reverse glycolysis? I mean, you saw the pathway, and a lot of those reactions are reversible. So why can't we just reverse it? Well, you can't totally reverse it. There are some reactions that can be reversed, and we use those in gluconeogenesis, but some reactions are too spontaneous to be reversed. So let's look at that. So here is kind of glycolysis and gluconeogenesis together. And you'll notice that in reaction 1, reaction 3, and reaction 10, the enzymes cannot go in reverse. And you can see the reversible reactions kind of have these two half arrows, whereas reactions 1 and 3 have two full arrows, indicating that we were using two different enzymes here. So different enzymes catalyze these steps. In fact, here, going from pyruvate to phosphoenol pyruvate, we have two steps to get there, whereas in glycolysis, we only have one step to get from phosphoenol pyruvate to pyruvate. So what is the key difference between, let's look at, we know what the key difference is in reaction 10. It's pretty obvious here. You've got one step versus two steps. But what about 1 and 3? What is the key difference? How would we know which way we were going? If we have fructose 6-phosphate, fructose 1,6-bisphosphate, well, how can we tell glycolysis and the gluconeogenesis counterpart? Well, you can see for reactions 1 and 3, it's basically the same thing, either glycolysis-wise or gluconeogenesis-wise. Glycolysis uses ATP hydrolysis to charge glucose to make glucose 6-phosphate, and to charge fructose 6-phosphate to make fructose 1,6-bisphosphate, whereas gluconeogenesis will hydrolyze phosphate and forms inorganic phosphate. Those are the key differences. And so here's a look at this. Glycolysis step 1, where we are hydrolyzing ATP and phosphorylating glucose. The net delta G is -4 kilocalories. Gluconeogenesis, where we are hydrolyzing glucose 6-phosphate with water to make glucose and inorganic phosphate. The delta G is -3.3 kilocalories. So the removal of ATP from the gluconeogenic side, using this hydrolysis of phosphate, is actually what makes the reaction spontaneous here. And we can imagine the same is true for reaction number 3. Okay, so now let's go on to the citric acid cycle, or the TCA cycle here. And so we take that pyruvate that we've made from glycolysis, and we can catabolize that even further into the very useful molecule acetyl-CoA, and that happens in our cells in the mitochondria. And acetyl-CoA enters the citric acid cycle. Again, a very important pathway in our bodies and in all organisms, really. It is also called the Krebs cycle, the TCA cycle, and again, for us, it's in the mitochondria. The citric acid cycle itself is circular. Step 1 is going to use the product of step 8 as a reactant. We'll see that when we look at the whole pathway. One turn of the citric acid cycle gives us, we start with acetyl-CoA, we end up with some GTP, which is guanosine triphosphate, we end up with carbon dioxide, we end up with coenzyme A, we had acetyl-CoA, we end up recharging NAD to make NADH, we have FADH2 when we started with FAD, and the net delta G for all of this is -11 kilocalories. And here it is drawn out with all the different intermediates. And you can see, so the product of step 8 is oxaloacetate. This then goes to become the reactant of the first step, where oxaloacetate plus acetyl-CoA yields citrate. And then the whole cycle starts all over again. It has to. If it stopped, we would be in a lot of trouble. So entry into the citric acid cycle is controlled, because of course you want to control things like central metabolism. You don't want it to go unchecked. So entry is controlled by that conversion of pyruvate, here's pyruvate, to acetyl-CoA, which is done by the pyruvate dehydrogenase complex. And you can see that several coenzymes are needed, not in the least coenzyme A, as well as NAD. So let's take a closer look at that. And you can see also that we need some FADH2 to transfer protons and electrons from NAD to NADH. And so here's a pyruvate. It gets attached to a molecule called TPP in the pyruvate dehydrogenase enzyme, which then gets transferred into this dihydrolipoyl transacetylase. And here it is here. It goes from this acetyl-lipoamide to, basically we get a CoA transfer and we end up with acetyl-CoA. The whole lipoamide thing gets reduced. It's reduced, it gets oxidized again, and it goes and does this whole thing over again. And the acetyl-CoA gets released and it's free to enter the citric acid cycle. Okay, so the pyruvate dehydrogenase complex, like a lot of other enzymes, is allosterically inhibited. It's controlled. Its activity is controlled. What controls it? And how are metabolic enzymes often controlled? That should give us the answer here. So it's controlled by the product, acetyl-CoA, of course. Too much acetyl-CoA that's not going fast enough, it's not going anywhere, it's going to come back and inhibit the pyruvate dehydrogenase complex. And again, this is another example of what we talked about last week in feedback inhibition, and what we're going to talk about again on Thursday. This is feedback inhibition. Metabolic enzymes being controlled by their product or by a product that's further down the metabolic pathway. And here is that electron transport chain again, and this would be in the inner mitochondrial membrane, where we have protons and electrons getting fed into these complexes. The electrons get shuttled along, protons get fed outside into the intermembrane space. This creates a gradient. This gradient, the protons are allowed to proceed down the gradient via the ATP synthase in order to recharge ADP and phosphate to make our ATP again. And you can see as one of the byproducts, where do these electrons go? Well, they go into making water. Alright, so putting all that together, what does the ATP yield from glucose? And depending on what book you're looking at, you can get different answers for this. So from glycolysis, we get a net of 2 ATP. And then we go into, basically we go into the TCA cycle, or the citric acid cycle. And then we go into the electron transport chain here. So from the citric acid cycle, we get a net of 2 GTP, which will equal 2 ATP basically. So far we have 4 ATP. Now, the big thing is the electron transport chain here, where we have NADH, 6 NADH here, 2 NADH from conversion of pyruvate to acetyl-CoA. We have 2 FADH2 from the TCA cycle. We have either FADH2 or NADH from glycolysis. And if they go down the electron transport chain, 6 NADH gives us approximately 15 ATP. 2 FADH2 gives us 3 ATP. 2 NADH gives us 5 ATP. And either we have FADH2 or NADH, which could give us 3 or 5 ATP for a grand total. And this is all from 1 mole of glucose, 30 to 32 moles of ATP. All right, enough fun with glucose. Let's talk a little bit about other metabolisms that occur in cells. So here is lipid metabolism, and lipids get broken down into their requisite fatty acids plus glycerol. Glycerol can be fed off into the glycolysis pathway here, and you can see how that might work here. Fatty acids then go into a special fatty acid degradation pathway, and they eventually will make acetyl-CoA. They'll eventually get broken down all the way to make a bunch of acetyl-CoA. So the first steps in lipid metabolism is hydrolyzing the lipid into its fatty acids and glycerol, activating the fatty acid with a group transfer. So we end up with a fatty acyl-CoA. We have CoA in the end. Moving the fatty acyl group across the mitochondrial membrane if you're a eukaryote. And then we carry out what is called beta oxidation, which we'll get into in a minute. Okay, so sugars are activated with phosphates from ATP. Fatty acids, on the other hand, are activated by coenzyme A. And ATP does play a role in that. As you can see, we do have to burn off an ATP and make AMP plus pyrophosphate to get fatty acyl-CoA. The activation is considered to require 2 ATP because we're hydrolyzing pyrophosphate in order to make this a favorable reaction, a spontaneous reaction. And so here's what one turn of the cycle would look like. We have a fatty acyl-CoA here. We, again using FAD and FADH2, we reduce a carbon-carbon single bond to a carbon-carbon double bond. Then we hydrate, sorry, not hydrolyze, we're hydrating that double bond to make an OH group here. We oxidize, and we get a carbonyl carbon here. And then we do a group transfer with a CoA, and we end up liberating an acetyl-CoA. And so the fatty acyl-CoA is shorter by 2 carbon atoms. So again, we are reducing a carbon-carbon single bond to a we're oxidizing a carbon-carbon single bond to a carbon-carbon double bond. We are hydrating the molecule, turning a double bond here into an alcohol group. And then we are oxidizing again to make a carbonyl, and then finally we're removing an acetyl-CoA. And so fatty acid beta oxidation is a spiral pathway. So we go through one turn of the cycle, we have a shorter molecule. We go through another turn, we have another shorter molecule. And here, in this illustration, you can see an 8-carbon fatty acid is being catabolized here, all the way down to acetyl-CoA. So it gets 2 carbons shorter every time because we're removing an acetyl-CoA. And we're getting a lot of ATP out of that if all the acetyl-CoA ends up going into the citric acid cycle, and all the FADH2 and NADH we make goes into the electron transport chain. You can see we end up with about 50 ATP. And that's even with the 2 ATP we use from the activation step. 50 ATP is pretty good. Now, why is it called beta oxidation? So I asked a question back when we talked about lipids. What are omega-3 and omega-6 fatty acids? And so you can imagine the answer for why is it called beta oxidation is along the same lines here. So recall the nomenclature of carbons on a fatty acid molecule, where on one end, the hydrocarbon end, the very hydrophobic end, that's the omega end. The carboxylic acid end is the alpha end. And so as you go down the chain, you're going from alpha to omega. So we start with alpha and then beta. And so omega-3 fatty acids, would have a carbon-carbon double bond starting at this omega-3 carbon here. One turn of the fatty acid beta oxidation cycle will cleave off an acetyl-CoA molecule from the beta carbon. So the beta carbon will end up being the new alpha carbon as we go through one turn of the fatty acid beta oxidation cycle. So fatty acids can be broken down. Of course, fatty acids can be made. They're made similarly in that it's a spiral pathway that we use. And again, we're adding carbon compounds as opposed to removing them. The carrier molecule this time, we're not using fatty acyl-CoA. That would get confused with fatty acid beta oxidation. We're using something different. It's called ACP. So ACPs are used in this fatty acid anabolism to make fatty acids that can go into our lipids. What is ACP? ACP is known as an acyl carrier protein. Fatty ACP is used for fatty acid synthesis or anabolism. It's similar to CoA in that it is a group transfer agent. Both have this phosphopantothene group to attach fatty acids. So this is ACP and this is coenzyme A. So this is how we're attaching our fatty acids. But one, it clearly has a protein part of it. This one is just an ATP on the end here. So there's the big difference between ACP plus CoA here. Both have their uses, and this sort of keeps everything straight. Coenzyme A is for beta oxidation, whereas the ACP is for synthesis. Amino acids, the catabolism, basically you get them amino acids through digestion of proteins and peptides. The important step is the removal of the amino group or transamination. This is involved in the catabolism of 11 of the 20 common amino acids. You can see a transamination occurring in both of these reactions here. We have alanine, alpha-ketoglutarate. We end up with pyruvate plus glutamate, or we can use aspartate and alpha-ketoglutarate to make oxaloacetate plus glutamate. And these pyruvate and oxaloacetate can then go into central metabolism. There's another way to deaminate amino acids. There's what's called oxidative deamination, whereas the deamination, I showed you the transamination, it just involves one amino acid getting transaminated to make another. This is just simply deamination, the pulling off of an NH3 plus group to give you ammonium, and in this case we got glutamate to alpha-ketoglutarate. So the amino group is replaced by a carbonyl group in this case. And so then it should be known that you can see the oxidation occurring using NADP, and then H2O is part of the hydrolysis of the whole thing. And when we do oxidative deamination, we're liberating ammonia. What's called the urea cycle will convert ammonia into urea, and you can see urea is a compound that has 2 nitrogens in it. Urea is is excreted from the body as urine, and so that is the nitrogen cycle in our bodies, where we have amino acids, they get deaminated, they come into the urea cycle, and urea gets excreted from our bodies. Deaminized amino acids could enter the citric acid cycle. You can see a lot of places where those happen. Alanine can enter when it's deaminated as pyruvate, aspartic acid as oxaloacetate when it's deaminated. Glutamic acid is deaminated to form alpha-ketoglutarate. Valine comes into succinyl-CoA. Phenylalanine, use your imagination on this, can become fumarate. And all of these can enter the TCA cycle to be used to make carbon skeletons or to make ATP, whatever is needed. For amino acid anabolism, it should be known that we as humans only make about half of the 20 essential amino acids for life. And so these take on a new meaning of the word essential. These are essential amino acids, meaning that we have to get them from our diet. So things like histidine, phenylalanine, tryptophan, arginine. These are essential amino acids for us to eat. And what do you notice about a lot of these amino acids? Histidine, phenylalanine, tryptophan. They're rather large, and they are rather expensive energetically to make. And so kind of over the millions and millions of years of evolution, we might have lost the ability to make all these different amino acids because it's just easier to catabolize our meat or our vegetables and pull out the histidine, the phenylalanine, the tryptophan from our food and just use it in our proteins, as opposed to having a de novo synthesis pathway for these things. Some amino acids can be nodes for further making of amino acids, further anabolism, and you can see that here. And we have glutamate. Glutamate can go on to become either proline, glutamine, or histidine, or arginine. Serine can become glycine or cysteine. Aspartate can become asparagine, or it can become lysine, methionine, threonine, isoleucine, and pyruvate can either become alanine or become leucine and valine. So you can see that a lot of anabolism of amino acids will produce more than one type of amino acid, depending on what node you're starting from, whether it's pyruvate, alpha-ketoglutarate, oxaloacetate, etc. Alright, that was long. So here's the summary. Metabolism is the movement and interconversion of molecules that are necessary for our survival. The goal for this is energy production. The goal is also to produce building blocks to make our biomacromolecules. Anabolism is the making of things like proteins, DNA, RNA, lipids, etc. Catabolism is breaking things down. The anabolic and catabolic pathways are often very similar. They often differ just either how you get from one thing to another, and obviously they differ in the direction you're going. Intermediates of different pathways can interconnect. Nowhere is this more evident than with amino acid catabolism or anabolism, citric acid cycle, central metabolism like glycolysis, fatty acid beta oxidation, things like that. Metabolic pathways can take on a variety of shapes. They can be linear, they can be circular, like the TCA cycle. They can be spiral, like fatty acid beta oxidation or fatty acid synthesis. Alright, so please read from the Raymond book. We're in the last chapter, chapter 14, pages 540 to 576. And I'll see you on Thursday, and we'll talk more about metabolism.